Yes, hi everyone. My name is Jemma. I'm here today to talk about a project I've been doing with Zoos Victoria over the last 12 months as part of the Wild Otways initiative. And this has been a massive program, as you've heard lots about today, and it's really involved lots of collaboration between the land managers and different agencies, the CEC. So thanks to everyone who's contributed information, data, support, conversations. It's been really great. So our project was investigating the feasibility of translocations of the swamp antechinus. Before I start, I'd like to acknowledge and pay my respects to the Victorian traditional owners as the original custodians of Victoria's land and waters. So in my project, I worked on the unceded lands of Eastern Ma for a lot of it, and also did a bit of work on Wadawurrung country as well. So I'd like to pay my honours to elders past and present. Some quick background about Wild Otways. We've heard lots about it already today. It was a federally funded project with a bunch of different investment streams. I think we've heard about almost all of these so far, which has been fantastic. Just to point out, we were working under the rewilding investment stream, and our project ran from June last year through to July this year. We've just wrapped everything up. I wasn't the only one at Zoos Victoria working on research in the Otways. I had colleagues that were doing surveys for broadtooth rats and smoky mice. Also, there was a project looking at the feasibility of translocations of leadbeater's possums as well. So there's been a lot of work down here over the last few years, and it's been fantastic to build on all the data that's already been collected. Some quick background on swamp antechinus. So this is a threatened species that's listed as vulnerable at a federal level. They have a distribution that covers Tasmania and mainland. There's a subspecies in Tasmania and on the surrounding islands. So we were really focusing on the mainland subspecies. You can see from that distribution map up the top, they're predominantly a Victorian species. So we should be pretty proud of this species and do what we can to protect it. Their distribution runs from down near the Prom and surrounding islands across to the border and just runs into a little bit of South Australia. And you can see the Otways sitting there in the middle of that distribution. But populations are now very fragmented across that range. Swamp antechinus occupy a range of different habitat types, everything from the wet heath to heathy woodlands, which we've heard a lot about today. But Barb kind of covered the fact that they are also present in a lot of coastal scrub, and some of those refuge areas along the coast are pretty important habitat for this species now. They have a generalist diet, so they feed predominantly on different insects, but they'll also eat small vertebrates, seeds and fungi. And the thing I love about antechinus is their crazy reproductive strategy. So they have this really interesting synchronous reproduction where for 2 or 3 weeks in the middle of winter, the males go absolutely bonkers trying to mate with as many females as they can before their testosterone goes through the roof and their immune system collapses and they all die at the end of the mating period. It's pretty wild. So that just leaves females and their offspring in the population. And the reason we think that this has evolved is that in the past, under very stable climates, there's this really predictable flush of insects coming into their environments in spring. So this really leaves lots of food around for the female and for her offspring. So you can see a couple of different females here that we captured, all the way from having tiny little tic-tac pouch young, size of tic-tacs, all the way through to bigger jelly bean-sized young that they're about to leave in a nest. So this makes swamp antechinus and other antechinus very susceptible to extreme events. So you might imagine if a burn or a fire comes through at the wrong time of the year, or if there's a drought that has an impact on invertebrates, these guys are really susceptible to that stuff. If a female loses a litter, unlike a lot of our other marsupials that can come back into estrus and breed again and produce more offspring, there aren't any males around anymore. There's no more males there for them to breed with. If a female survives to the next breeding season, and many of them don't, she needs to wait for that cohort of pouch young to become sexually mature and those males to enter the population for breeding. So you can see that for a species like this, things could change very quickly year to year. So as for a lot of our other small mammals and medium-sized mammals, they're threatened by things like habitat loss and fragmentation, changing fire regimes, talked a little bit about climate, things like drought, but also Barb touched on the issues that we're seeing around predicted sea level rise, particularly in some of these refuge habitats that are, expected to possibly disappear, these habitats, in the next 5 to 10 years. But also introduced predators. We've heard a lot about foxes and cats today, and a small species like this is particularly vulnerable to predation by cats. So our project aimed to investigate if translocations were feasible for the species. It entailed a lot of work, and I've put lots of words up here, so I apologise in advance. I'll try to run through them pretty quickly, and then I'll just focus in on a couple of points. But we started our project on the run with 2 months of winter trapping in the field, with the aim of collecting some new genetic samples and looking at the distribution of the species in more detail. So we worked all the way from the Anglesey area down through Cape Otway and to Port Campbell region. So at the end of this season, we were able to collect some new genetic samples. We were also lucky enough to get our hands on some historic genetic material from Deakin Uni and the museum and from some other collaborators. And this allowed us to undertake a comprehensive genetic analysis for this species using more modern techniques than what's been done before. We worked with First Nations groups, particularly Eastern Ma, to get their perspectives on translocations. And we also ran 2 workshops on different translocation scenarios with a number of land managers, some of whom are in the room today, and other stakeholders as well. This led to us assessing 4 potential translocation sites where we might think about moving swamp antechinus if we were to do a translocation. The first was Wildlife Wonders, which is managed by the CEC down near Apollo Bay, and that's the only fenced location in the Otways region that we thought might provide a good opportunity to have an insurance population of swamp antechinus in this current area. We also looked at a couple of sites outside fenced areas in the Otways National Park and the Otways Forest Park. We've heard a lot about Carlisle Heath today. It's also a very important place for swamp antechinus, and that was a potential source site that we thought might be suitable due to the high abundance of animals that had been trapped there in the past. So we carried out some assessments using cameras and trapping to look at whether or not that would be a suitable place to source founder animals from. In 2023, we did some autumn surveys in the Carlisle Heath as well to start to monitor population trends in the west. We've got some amazing data that Barb talked about this morning from the eastern Otways, but we don't have great monitoring for small mammals or swamp antechinus in places in the western Otways. So we've sort of started that by going back to sites that were trapped out in 2021 and starting to build up some longer term reassessments at the same locations. At the same time in autumn 2023, we undertook some field trials looking at different monitoring technology. So our intentions here were to look at whether or not we could non-invasively monitor swamp antechinus after translocation. So could we use microchip readers set up on supplementary feeding stations to look at how they're doing post-translocation rather than having to rely on trapping animals. We undertook the first disease screening for this species. So we did some cloacal swabbing of animals that we captured and have worked with the vets to do the first microbiology assessments in this species. We got all the species experts together and we ran a workshop to prioritise conservation actions for swamp antechinus over the next decade. So that's been a really valuable exercise that's been a great addition to the Wild Otways project. And then finally, in the last couple of months towards the end of the project, we've been collecting additional genetic material and specimens that have turned up after the male die-off with the aim of doing some more genetic analysis in the future. And we managed to do some surveys also outside of the Otways. So we went to Bunyip State Forest in State Park in Eastern Vic, back to a site that hadn't been trapped since the 1980s. And we also did some surveys on the Mornington Peninsula. So because we don't have much time today, I'm just going to focus on 2 of these components, just like quite quickly, looking at assessment of translocation sites and our genetic analysis, just to give you a bit of a feel for how we went about that. So for our release site planning, our objective was to identify and assess the suitability of some of our proposed translocation sites. To do this, we used IUCN guidelines. So we looked at things like habitat suitability, climate requirements, estimates of carrying capacity, what types of threats were at sites and how we might manage those, and also whether or not there were other terrestrial mammals or conspecifics present at those locations. To do this, we did a bunch of habitat surveys and also undertook some camera trapping. We also used a bunch of different habitat models and tools that are available, and that just helped hone in on some places. We used habitat distribution models, so you can see an example of one of those here. The dark purple areas are more suitable habitat compared to the paler areas. So these models are great at a landscape level of kind of identifying where places might be suitable due to their climate or terrain, but you really need to get out on ground to check them. So, for example, there's some areas through that model that are actually agricultural areas now that have since been cleared. So it just points you in the right direction. Another useful tool is ecological vegetation class mapping. So we also used that to identify places that had similar EVCs to where we'd found swamp antechinus in the past. And then we went to those locations and also mapped the habitat on the ground, looked at the dominant plant species that were there and made sure that they were similar to other places that you would find swamp antechinus. So that allowed us to make a few recommendations about translocation sites into the future. One of the other key points was that carrying capacity was really important. So the size of the area and the ability of that area to support a large number of animals also played into our recommendations. I'll quickly move on to the genetic analysis. So I'm just going to present results today from our new genetic data. I'm not going into any of the historical data that we analysed, just the data that we've collected from the last couple of years. We found that there were significantly differences in genetic diversity between different populations of swamp antechinus. So from this figure up here, all you really need to take from it is that each dot represents a different animal, a sample. We've just taken a small ear tissue sample from those animals. And the different colours represent different populations. So right at the top, we've got some samples from Wilson's Prom in the yellow, and they were kindly collected by some colleagues of mine. On the far edge, those pink samples are from Cape Otway, and then the rest of the Otways is kind of shown in that middle bit. So once we exclude those sort of outlying samples from the Prom and Cape Otway and zoom in on these this clump of samples, you can actually see when you look at them more closely, there's no mixing of those colours. They're all different populations. There's no genetic mixing really between any of them at all. They're sitting quite separate to each other. It was quite interesting that Cape Otway is sitting really differently from samples that were taken east and west of the Cape as well, possibly suggesting it's been isolated for quite a long time. One of the other things that we found was that the swamp antechinus in the Carlisle Heath, that was the only population that we had enough samples to look at things like inbreeding metrics, but they had significant inbreeding values and low relatedness. So that suggests that in the Carlisle Heath area, this is a signal of a past bottleneck. So the numbers in the population have dropped to really low levels at some point in time. We can't actually track exactly when that is, but whether or not that's due to a drought or due to the 1983 fires, some disturbance in the past has meant that they've come back from a really small number of founders. So this is the most comprehensive genetic data set that we have for this species now, and it's really helpful for informing what you might do if you were going to do a translocation in the future. So we know that we wouldn't just take animals now just from the Carlisle Heath due to those inbreeding metrics. We would want to mix those with animals from other locations. So places like the Warrnambool Plains, sites through the Warrnambool Plains and around Colac Creek are all suitable places, but you would create a founder group from a mix of animals in these locations. The other thing we need to do is really focus our efforts in getting some more genetic samples from some of those coastal refuge populations. And Barb really highlighted, the importance of those places this morning and how vulnerable they are to sea level rise. We don't have great genetic data from there now, so it'd be really good to get some understanding of what's going on there and then look to the future of maybe doing translocations, moving animals out of those vulnerable places to other locations. Every little bit of genetic diversity is really helpful to preserve into the future, so it would be a shame to lose animals from those refuges. So that's about it from me. I'd like to thank everybody who's worked on this project with me, had discussions, the whole Wild Otways team. It's been a really amazing project to be part of, and I feel very privileged to have worked with all of you. So thanks for listening, and I'll take some questions.